Okay, well, thank you for the invitation. So my name is Peter Kharchenko. I'm from Harvard Medical School. I have a small group, mostly doing computational things. And for the past number of years, we've been looking at single-cell genomics methods, mostly functional genomics. Initially, I was going to talk about applications to cancer analysis and the sort of the problems that occur there and what we're trying to tackle. But yesterday's talks actually inspired me to give you a couple of vignettes instead that touch on genomics, on genetics, in hopes that they will stimulate some discussions and potentially new methods. So I'll give you a brief overview of some of the things we're dealing with in terms of single-cell RNA-seq, and then try to give you these couple of vignettes. All right, so first of all, just, we're back to single cells, and we had a couple of talks today, but I wanted to add some motivation for all this. I think most of our knowledge about biology and genomics in particular comes from these bulk samples that contain millions of cells typically. And the knowledge about individual cells really comes from microscopy field for the most part, with flow cytometry and a few other methods. And only in the recent several years, basically, we started to learn new biological insights from sort of single-cell genomics methods. If you think about, these complex tissues, they're all basically composed of very different cell types, usually, that in combination give rise to the overall phenotype of a tissue. And to even understand all the components and how they interact with each other, how they support each other, that can be quite a bit of work. For instance, this is the hematopoiesis. And this type of a tree or structure is a result of many, actually decades probably of efforts of different researchers. And then here on this slide, this is a single measurement. It's a single cell measurement, but a proteome-based one of mouse hematopoiesis. And the labels here are based on some of the known surface markers, but basically once you put the labels here, you can more or less try to follow this tree and learn a lot of very important things about hematopoiesis from a single snapshot. So every point here is a cell that was measured, and they're arranged relative to each other, so cells with similar combinations of markers appear nearby in this 2D projection. So they form this type of trees, and you can basically read and try to get new insights from this. Okay, so overall, what types of things, different things can single-cell genomics, single-cell analysis get you? Well, the main thing you get, because you're measuring individual cells, you're keeping track of this, is variability between the cells. You can start to actually quantify that. So sample composition in a rough sense. You can learn about stereotypical subsets of cells, whether they're cell types or cell states. If you're measuring genetics, for instance, that means looking at genetic subpopulation, subclones, for instance, that occur in cancer tissues, sometimes in normal tissues. The important thing here that particularly functional states like transcriptional state, epigenetic state of a cell, they're very high dimensional, and they can reflect a variety of processes that influence the cell. Obviously, the cell type will influence, for instance, transcriptional profile, but so will the cell cycle, so will its microenvironment and epigenetic state, and so on. And what you see in many of these measurements is a combination of all these influences. Some of them are much more prominent than others, but it's important to keep in mind that there are probably multiple signatures, sort of independent signatures that occur per cell, with some interactions on them. The other thing you see are gradual differences, right? So instead of sort of a discrete subpopulation units, you can have trajectories, you can have kind of elongated clouds that reflect sort of gradual response to some external stimuli and so on. The other sort of aspect, flip side of it, you could start looking at the covariation within the individual characteristics you're measuring. So let's say if you're just measuring transcription, you can look at correlation between different genes, right? Either between subpopulations, which is probably better expressed in terms of subpopulation structure, or within subpopulation that might give you some insights about the regulatory process underlying it. If you're doing multimodal measurements, if you're measuring, let's say, transcription epigenetics at the same time, you can start looking at correlation, let's say, between gene activity and transcriptional level and the epigenetic state and so on. All right, so from single cell to genetics, and this is just kind of a conceptual slide just to give you some ideas, and then in the vignettes I'll show some examples of it, which are obviously still quite fresh. Okay, so what is single-cell analysis? How can it contribute to kind of genetic studies and studies of genetic variation? So I think conceptually one thing it can do is give you sort of a partitioning or segmentation of a genome by function, right? So genome is very large and it's hard to find associations in there. If I told you somehow that only these 5 spots are somehow relevant to the process you're actually interested in, that it's a huge improvement. So what these single-cell methods, especially functional methods, may allow you to do is to do sort of in silico separation of different cell-specific effects. So first of all, let's say you have types of cells that you're not actually able to isolate from the tissue physically, and that's a pretty common thing. But you're able to distinguish them in these single-cell measurements. Then you can group these computationally and come up with, let's say, aggregate expression profiles or epigenetic profiles or something like that. So it gets you, these in silico subpopulations that gives you cell type specific activity profiles, let's say, expression and so on. And if you're running EWAS or epigenetic association, you can actually use those without explicitly physically purifying the cell types. Again, just to repeat, sort of what you can try to do then is try to focus, let's say, your association study on the relevant parts of the genome. So for instance, if you know that a particular cell type involved in pathogenesis, then you can focus on the regions that are active, genes that are active, regulatory regions that are active in that particular cell type, and narrow down the scope of that search. The other thing you can do is actually try to guess which cell types may be relevant. If this is a very complex process with many cell types participating, you can try to figure out which cell types are more likely to mediate the progression of the disease. The other thing you can do is look for genetic variation within cells, right? So these are a little bit harder to do. So we heard yesterday about genetic mosaicism in normal tissues, and if you're measuring actual genes, if you're getting some sense of the sequence, you can start looking at that. These are typically rare events, and so they will be hard to detect within the noise. One place where you have quite a few events is within cancers, and so there intertumoral heterogeneity is fairly extensively studied, and you can look at subclonal populations and other sort of genetic variations within cancers. And at that point, you can start looking at basically variation within a population you've already measured. At the moment, technologically, it's very difficult to measure wide panels, including many patients. So we're talking about dozens, really, samples realistically now, maybe up to 1,000 in the next couple of years, but it's still a very expensive technique. All right, so techniques, for the past few years, there's been a lot of progress in RNA sequencing, specifically mostly due to improvements in microfluidic handling. Fluidiome machines were quite popular a few years ago. One of the first microfluidic devices for single-cell, commercial devices for single-cell RNA-seq, basically based on these kind of physical traps. This is a cell here in this one trap. You close the valve, slice the cell, prepare a library, and then there are about 96 traps like this on that chip. In parallel, there are methods based on plates where you sort out individual cells into wells and then prepare the whatever library you want using a protocol of your choice. Lately, these microfluidic, droplet microfluidic measurements have been very popular. I'm not going to play the video, but basically you have these droplets flowing together with encapsulating individual cells together with beads that carry different barcodes that will allow us later on to recognize the material coming from that particular cell. All right, so this is kind of an advertisement slide, but basically just to emphasize the amount of growth in the data that we had here. This is comparing with Moore's law, of course. These are the CPUs I had on my desktops over the years, and if you just line them up, the number of processors, it does work. It's a pretty linear sequence on a log scale. Now this here is more interesting. So the counts here are numbers of cells in sort of landmark studies that happen over the past 5-10 years. And initial studies, we're just talking about a handful of cells. Very nice papers from Azim Surani's lab that looked at early embryo, and that was really state of the art. First kind of a whole genome, high throughput measurements based on START protocol from Stan Linderson's lab. It got us to about 100 cells, and then very rapidly this is droplet microfluidics, and now we're talking about millions of cell data sets that are being released. So this kind of creates problems because basically any method you develop today will have to handle an order of magnitude more data tomorrow. or next year realistically, and that's quite tough to do. So these days, if we're designing a study, basically we're talking about hundreds of thousands of cells typically. In the transcriptional measurement, we get about 10 to the 4th cells. Even in sparse format, it's still pretty large data. More concerningly, I guess, in terms of method development, now that these methods have matured, people are transitioning to sort of actual realistic studies instead of kind of pilot demonstrations. So we're talking about case and control panels, we're talking about multi-patient cohorts that are being measured, and all these require additional statistical considerations to actually analyze. So I think it's kind of right now a combination of both a computational task, simply being able to make sense of this data in an efficient way, and a statistical treatment at these different levels. I'll show you a little bit just on the lowest level, the measurement itself. This is a fairly old slide, but I think principles are still true today. So we're comparing expression of different genes. Each point is a gene between two different cells. And let's just imagine cells are identical for a second. If they were identical, you see everything along the diagonal here, right? But you see some kind of a spread here. So the spread, you would see this in a normal RNA-seq and a bulk RNA-seq measurement as well, but it's typically wider in the single-cell measurements, so high over dispersion. You tend to get outliers that are hard to explain with almost any statistical process. You also get these kind of streaks along the axis where we've seen a gene expressed at sort of moderate expression level in one cell and not at all in another cell. So this reflects both the sparsity of the measurement as well as these excessive dropouts that can happen due to library preps. Data basically is count data, right? So all this is distributed, and it's not very Gaussian basically unless you transform it and normalize it properly. And if you simply apply methods like PCA and so on, when the transcriptional differences are more subtle, the technical variability can mask the actual biological variability that you want to see. So distinguishing kind of bad cells, for instance, from the ones that have been successfully measured and so on. All right, so a couple of things we did for this is basically try to derive more accurate noise models for individual cells, and then come up some kind of variance rescaling based on that. So the basic noise model we've used is a mixture model of two simple processes. So you have, for a given, let's say, gene in a given cell, we either convert it through RT and amplify it, and then the responses in terms of counts is something like negative binomial distribution, or we fail to do so, and then our counts are basically low level, some kind of background post-home process. So this mixture fit can be performed on individual cells, given that we have some kind of an expectation about expression magnitude of a given gene. The model doesn't have many parameters per cell, about 4 or 5 parameters, and the expectation about expression magnitude you can get by basically looking at the sort of neighbors, cellular neighbors of a cell. And that gets you, in this case, if you look at this middle plot, in blue shown the cells that are much more likely to be, the genes are much more likely to be in part of this negative binomial component, and the red are the dropout events modeled by Poisson. The frequency of these dropout events depends obviously on the expression magnitude. For highly expressed genes, you almost always detect them in a cell. But for medium low expressed genes, this is where we see more and more of these dropouts. So I'll just show you a few examples of these, and I think you can mostly focus on the central plot here. This is a slightly updated model, also shows in these black dashed lines the 95% confidence band. And again, this is starting with Fluidigm C1, a slightly older technology. As you can see, there's a lot of noise, a lot of spread here over dispersion. The genes that we do detect are kind of located here, compressed in this one region. So, in fact, the dynamic range of detection is very much compressed. And there are actually a lot of zeros here. The excess of zeros doesn't look that bad, but this is on top of already very large overdispersion in the negative binomial component. This is one of our in-drop measurements, so one of the droplet microfluidics methods, and you see sort of more linear response here in terms of counts detected in this particular cell. You also see these discrete levels, and then that's basically discrete counts. It's a very sparse measurement, so you detect relatively few molecules per cell. And the dropout frequency here, the excess on top of this negative binomial, is quite significant, up to 60% or so. So it gets you kind of high confidence measurement, but which is very sparse. This is a SmartSeq2 measurement, another method that actually does full transcriptome, full length transcript coverage. It's a plate-based method that tends to be more expensive and low throughput, but it can produce very nice results. So here, for instance, you do see also very kind of linear response, which is almost surprising given the fact that they don't incorporate unique molecular identifiers, so any sort of corrections like that. It's just very clean amplification. You also see a lot more genes detected, so it's denser in this regard, and the fraction of these unexpected kind of dropouts is pretty low. Just to get you a sense for the amount of noise per cell. Okay, so we can get a better handle on technical noise by basically describing it using some kind of a model. And the first thing we'd like to do really is characterize, what do we have in this kind of a mixture. Given this kind of basket of apples, we'd like to know, what types of apples are there, green, red, if they're rare apples, we'd like to know that too. We also would like to know, what characteristics actually distinguish them. And so to detect subpopulations, especially in kind of small samples, we wanted to use some prior information about genes working together. So we actually went with analysis of gene sets, both pre-annotated and the ones we detected from the data. And the idea is pretty straightforward. It kind of parallels with gene set analysis that you typically see in the differential expression analysis and things like this. So essentially, if you have, let's say, 5 genes that are all together up, 5 genes that are related, let's say, to a particular known biological process like actin upregulation, and these genes are all up in one subset of cells and all down in another subset of cells. First of all, it's much easier to detect, right, given a very noisy measurement, and it also gives you some kind of a hint as to what the biological process underlying these 2 subpopulations might be. So that's the general idea. Now, in terms of statistical treatment, basically what these individual per cell models allow us to do is to score the variance accordingly. Now, a major confounding factor here in the variance is actual expression magnitude of a gene. So this is a coefficient of variation squared on log scale, and this is expression magnitude, sort of average expression magnitude of a gene in the entire population. You can see a very strong dependency here. Now, you could try to model this analytically, but it turns out that, depending on the protocol you use and the measurement and so on, this curve can look slightly different. So there are probably a lot of factors contributing to this, so we just use some kind of local regression fit to fit this overall trend. And what we're doing then is using these mixture models, so negative binomial Poisson mixture models per cell to estimate the amount of basically statistical confidence in the variance that we have, separation from this genome-wide trend, and it basically tail probability of that process translated back into some kind of very simple, chi-squared distribution. So that allows us to basically rescale the variance of these genes to put them in some kind of uniform footing. So when we apply, let's say, PCA or some other analysis, the relative contribution of different genes to this analysis will be appropriate. In this particular case, we're looking at gene sets, so we're hoping to find cases like this where genes from a given gene set are all up, let's say, in some subset of cells. And we're looking at these genes that are varying more than we would expect, right? So over-dispersed genes. And obviously, if a gene is differentially expressed in different populations, it would have higher variance than you would expect by chance. So the basic idea is that we're doing these weighted PCAs. The weighting is just to account for the frequency of the dropouts. And we're doing these weighted PCAs per gene set. So we take a set of genes, so every column is a cell here, every row is a gene, and this particular set of genes shows high expression in these 2 cells, for instance. And so first principal component, our projection on the first principal component would basically give us something like this. This is the orange-green pattern here, separating these cells from everything else on the data set. And we can do this for different gene sets, obviously, that we have in some database. Now, we can score the magnitude, basically the amount of variance that this principal component explains. And if it's statistically significant, then we'll take it into account into further analysis. We'll basically try to visualize and interpret it. Now, obviously, the annotation that we have, like gene ontology or some other gene annotation, can be incomplete. So we also detect a set of genes that are simply correlated within the data set that we have. And that gives us another set of gene sets, and so we can repeat the same procedure here, except that because we specifically pick them to be correlated, the statistical scoring of these principal components has to be done using different bases. But overall, we can use essentially random matrix theory to get the scoring of these of these principal components. So after this, we get kind of a collection of basically patterns that are separating some cells from others. And a lot of them will be very redundant. If you ever worked with gene ontology, for instance, there are a lot of very similar gene sets. And also, if there are, let's say, 2 subpopulations of cells in your data, they might have multiple processes, biological processes, that are actually separating them together. We put this all in this kind of a browser interface. I'll show you in a second. And the reason for this is because basically the interpretation of these results, whatever statistically significant patterns you find, the interpretation of them is still kind of a manual art at this point. We don't have a good way of annotating the biological functions of different subpopulations. And so we need to have a convenient interface to actually look at it. This is a result, again, columns of cells here, and these are cells from mouse, hippocampus, and cortex. And what I'm showing here on the as rows are these aspects of heterogeneity, basically in orange and green, separating some cells from others. And we're just listing them here as rows. And if you look at different rows, for instance, the top 2, you can see the genes that are basically driving this separation. So for instance here, we have expression of these innate immune response genes, and these cells are actually separated because of strong expression of myelination pathways. And so you can go through these and actually try to do an annotation of some sort. I'll show you a quick example, but so this is the same result, basically. This is just kind of a 2D, oops, you can't see anything. 1 second. Yeah, so same result. This is just a t-SNE representation. So if you start kind of clicking around, you start seeing both the gene sets that actually gave rise to this particular pattern, main genes that are separating this subpopulation of cells, in this case here, and so on, and try to look up additional genes, try to actually make sense of these subpopulations. So these types of tools are actually quite valuable. Now I have to close this somehow. Wow, this is really difficult. All right. These types of tools are quite valuable for trying to make sense of why certain subpopulations of cells are actually being separated. Now, as I mentioned, the scale of the data has been growing considerably, and so now for these kind of newer data sets, so we're talking about hundreds of thousands of cells potentially, we needed different methods. We've designed this second generation package, Pagoda 2, which is optimized for these large measurements. Again, doing some kind of filtering here, variance normalization based on a slightly simpler statistical model. And then it goes through the same procedures to identify basically subpopulations of cells. So that's kind of clustering procedure, visualize them somehow, so you can actually point at the 2D picture and talk about subpopulations, run differential expression, figure out different sort of cross-cutting aspects by which these subpopulations separate, and so on. Now the real advantage of this, that this is a video that ran. But it ran in real time, and it's about 33,000 cells and completed in about 20 seconds. So we basically try to optimize it for speed so we can build more advanced analysis on top of this, including analysis of much larger cohorts of samples. The pathway analysis, the gene set pathway analysis is still there, although I think nowadays we get such amazing reference data from the single cell data sets themselves that the sort of prior information, or the value of the prior information we used to get from the gene sets, it doesn't seem to be very significant anymore. So we're mostly switching to these types of reference-based analysis I'll show in a second. It has a slightly updated interface, allows you to do more things like differential expression on the fly and so on. All right. Yeah, this is just a quick example of making use of these reference data. So here, we actually have prostate cancer samples. from one of our studies, and we projected them in a way that takes into account both the axis of variation that were found within this particular data set, as well as axis of variation, the transcription variation that we've seen in the reference data set. And then we can project the reference subpopulations onto the subpopulations in our data, just to get some kind of rough labeling of our data set, so you don't have to go through interpretation again. And in this case, the reference was blood, so you see everything but these cell types labeled, and actually these cell types are tumor-specific cell types, some actual tumor cells, and then some stromal microenvironment that tends to co-occur or expand within the tumor fraction. The last thing that, as I mentioned, there's this issue of how to bridge together panels of many samples, and for this, we're developing separate algorithms that are basically based on very large graph analysis. Okay, so first vignette is about heterogeneity in the human brain. So this is a collaboration with Kun Zhang and Gerald Chun here in California. And the idea is that we sampled a number of brain regions and then did measurements on them, so that you can't really isolate individual neurons from adult human brains. Very difficult. You can, but you'll probably lose the vast majority of them. So what we're isolating here are nuclei, and we're measuring a transcriptome of the nuclei. Now, obviously, it's a subset of all molecules that are actually in the cell, but it can be quite informative about the type of the cell. In parallel, so this is actually one of those single nuclei expression data sets, and again, I'm just showing these t-SNE plots to show different subpopulations of cells that we see, excitatory, inhibitory neurons, and oligodendrocytes, and so on. Now, the interesting thing about this study is that in parallel, we also measured chromatin accessibility at a single cell level from the same samples. Now, we're not measuring the same cell here. The cell, every time gets destroyed, but we're measuring the same, basically, mixture of cells. In parallel, we're using two different assays. And this particular one is, well, I'm sure you're familiar with this. There was a talk yesterday, basically, but it looks at accessibility of the chromatin, and what you see typically are accessible regions within open chromatin that quite often marks regulatory regions, promoters, and so on. It's very sparse, right? So per cell, obviously, you can have many copies of a transcript, but you only have a couple of copies of the DNA. And so whether the cut occurs or not, if it occurs once, it pretty much saturated the signal in this case. And then the number of cuts that you can get overall per cell is not that large. So here, this is just a part of the genome, and every row is a cell. I've already classified them somehow, but just to show you the density of the signal. So for instance, just increased occurrence of cuts within this region would be separating endothelial cells from something else. So it's a very sparse signal spread out across the genome. Now, you could do the same thing basically on this side for transcriptional subpopulations, and this is for epigenetic subpopulations. So we used a slightly different model here to account for this very early, very rapid saturation of the signal in this chromatin accessibility assay, basically a censored Poisson model. But you also can cluster cells and group cells based on the similarity of the overall chromatin accessibility profiles, and you get some subpopulations here. Now, an interesting thing, and somewhat unexpected to me, that basically the complexity of subpopulations we're seeing from a epigenetic standpoint is actually lower than transcriptional standpoint. And earlier studies on bulk analysis, from ENCODE and so on, pointed out that the epigenetic state of, let's say, DNase I hypersensitive sites, of regulatory regions in general, is much more dynamic, and so you would expect more distinguishing signals in that mode of measurements than transcription alone. But I think just the measurement is very sparse and noisy, and that's probably why we're not seeing separation to the same extent. But we're measuring the same population, so we can first of all try to assign the labels consistently. Please.
So if you measure fewer cells for chromatin accessibility, would the resolution be better of these data?
No. So, and again, it depends on what you can do. But there are a lot of kind of technical inefficiencies that I'm sure can be improved. But in principle, the way these assays work is that you have a bulky enzyme coming in and making a cut, right? And so it can only make a cut once. And so you don't actually have a good sense for how confident it was when it was trying to make that cut. So there's very limited amount of information that you can sample essentially from an individual cell, from an individual locus. So you're probably much better off measuring a lot of cells instead of fewer cells. But obviously the specificity of that enzyme in terms of accessible versus inaccessible region is also very important, and that can be improved. That will give you slightly better signal-to-noise ratio. All right, so we came up with this kind of iterative strategy to basically map between transcriptional and epigenetic populations, and the idea is pretty straightforward. We don't know in general, we're pretty bad so far at predicting which, let's say, regulatory sites are talking to which genes, right? And the other way around. But we can try to guess. So, typically the regulatory regions within the promoter proximal, for instance, regions are somehow influencing the expression of that particular gene, right? Same thing, if you have, let's say, an enhancer within an intron of a gene, it's quite likely to be influencing that gene, although less so than the promoter region, and so on. So we put in all kinds of predictors, very technical predictors like distance to the gene, position within the gene, and so on, to basically try to learn a kind of predictive model that would in general give us some kind of a weight saying that if this particular gene has changed, how likely we are to see a difference in the regulatory site that is somewhere nearby of that, close to that gene. And so what we're trying to do here is kind of the following. So we can go between transcription and accessibility in both ways. So I'll just describe going from transcription to accessibility. So let's say if we know transcriptional difference between two subpopulations, astrocytes or oligodendrocytes in this case, if we already know a set of genes that's supposed to make them different, we can try to predict which set of enhancers, for instance, or regulatory sites would be different between them. And if we try to do this type of train these models, this is a gradient boosting model that we use here. If we try to train these models and try to predict change in accessibility of an individual hypersensitive site, we'll do really poorly, right? So just overall, the regulation is very complex. Our ability to detect the change is probably not absolute, and so we're not doing well. However, we have many regulatory sites that we basically expect to change, between very distant subpopulations like this. And so when we combine them all together to classify a cell, whether it's, let's say, astrocytes or oligodendrocyte, then the joint probability is actually very, very strong discriminator. So we can do pretty well in terms of classification. So what we're doing here is we're looking at this transcriptional taxonomy that we just derived based on RNA-seq data, and we slowly walking down, traversing this taxonomy. So the top split here would be between neuronal and non-neuronal cells, right? That's the most striking transcriptional difference that we have in the data set. And this is the view on the transcriptional data. And if we go through this kind of model learning and then prediction and evaluation of that classification, we find an epigenetic signature that clearly separates the two, right? With very high confidence. So that's easy. We could probably tell that by eye anyway. So, and you can then go down to a second split, for instance, between excitatory and inhibitory neurons, and repeat the same procedure. You'll still get a very strong score, and keep going down, further and further splits. So between, let's say, two subpopulations of inhibitory neurons, we're not doing so well anymore, but we can probably distinguish them still. And if you go into even finer subclassification, we basically stop getting signal there. So overall, in this transcriptional taxonomy, we can go down to a certain subset, to a certain level, basically, to distinguish subpopulations within the accessibility space, but not below that. We can also refine these signatures and basically try to get a better kind of subpopulation classification within the chromatin accessibility space. So that's kind of just putting these two data sets to sort of views of the cells on the same footing. Now, all this work can yield us very simple looking results. So we can basically take, let's say, all the cells that we have classified to be excitatory cells of a particular subtype, right? And simply pool all the accessibility data for them, and that will give us a track an accessibility track that looks very much like a bulk measurement, right? And we can do this for any of the subtypes that we've identified within accessibility data. And then you can start doing things that people have done previously for these types of data sets, look for overrepresentation, let's say, of transcription factor binding sites and so on. That's actually, that's an example here. Different cell types and different transcription factors and overrepresentation. More interesting, again, going back to the idea of connecting this with genetics, we can try to answer that question as to, what cell types appear to be most relevant to different types of disease. And in this case, we're making use of significant GWAS hits and simply looking for overrepresentation of these GWAS hits within the accessible regions for different cell types. The scoring is based on some kind of randomization procedure. We basically take pretty wide regions, about 50 kb regions around each GWAS site to score this, and simply look for overrepresentation or significance of overrepresentation. So here rows are different subpopulations, sort of a crude classification, and then columns are different diseases. We include some controls like type 1 diabetes. These are sort of more brain-related diseases, Alzheimer, Parkinson, and so on, where we expect to see, for instance, higher significance of excitatory neurons, inhibitory neurons in some cases, and so on. Now, one thing to emphasize, we're not trying to infer causality in any way. It's a purely kind of mechanistic relevance. So what we can say is that, if there's a highly significant z-score here, that somehow that cell type most likely mediates the progression of the disease. Now, whether it's actually causing it or simply making it worse, we're not sure.
Sorry, I missed.
Please.
How do you compute the z-score for a specific subtype?
So what we care about is a signal where GWAS hits for a particular disease tend to occur preferentially near open chromatin regions specific to a particular subset of cells that we've detected, right? So we look for, we segment these regions, we basically count the number of occurrences of these GWAS hits, and then we score this against the background distribution. The background distribution is simple randomization of the genome.
Does this include LD?
Yeah, so the regions are pretty large. We've considered randomizing by LD blocks, but it didn't actually make a difference. So we don't explicitly include it in randomization in this case.
So this is not from single cell, but this would be done for GWAS that was specific to that cell.
No, so GWAS, it's germline genomes, right? So it's probably from blood samples and so on. All we're using from the GWAS studies are the significant hits, right? So all the information we have is that this set of positions, now markers anyway, is associated with a given disease, right?
So you use the cell types to infer what regions are accessible, and then to use those accessible regions, GWAS hits.
Yes, if you had, let's say, Parkinson's disease, and you had a particular set of risk alleles there, and you find that these risk alleles tend to be within the accessible regions of, let's say, excitatory neurons of type 2, but not 3, right? That's the type of inference we're trying to do. Let me go to the next slide. It might be a little bit easier. So here, for instance, again, the bluer it is, the more significant it is, and this is schizophrenia, and we're just looking at a subset of these excitatory neurons. And it's pretty evenly distributed, even though there's a enrichment for excitatory neurons in general, but it's evenly distributed across subsets of excitatory neurons. Whereas with Parkinson's disease, it's quite localized to this particular subtype of excitatory neurons, right? So it's saying that the whatever mechanism is, somehow it appears to have more relevance here in terms of activity of the cell. Okay. How much time do I have? Until
2 minutes.
2 minutes. All right. Not much I can say about this. Well, let me just give this kind of motivation overview, and I'll skip the method itself. So the heterogeneity, genetic heterogeneity in cancer is a huge problem, or heterogeneity in general, right? Because essentially, when we have a drug that we use, it typically kills a huge percentage of actual cells it's supposed to target, but let's say 10% survives, expands, and then that's how you get recurrence. So cells can also develop drug resistance. And the best studied aspect of heterogeneity is genetic heterogeneity, so occurrence of subclonal mutations, subclonal populations, and it's been studied extensively using bulk genome sequencing and more recently using single cell genome sequencing. And what we tried to do here is look at relationship between genetic and transcriptional heterogeneity. So the transcriptional heterogeneity it can be quite apparent if you simply measure transcriptional states of cancer samples, and you can imagine a situation where each clone has its own transcriptional signature, right? That is quite distinct, and that's all the transcriptional differences that you see among cells. However, you could also imagine a situation where transcriptional differences are driven by other processes other than genetics, by the microenvironment in which the cells find themselves, epigenetic state, and so on. And in fact, those differences may be more impactful in terms of ability of these cells to resist treatment or not. So what we tried to do here is to look at the RNA data, essentially expressed exomes, as you could call them, and try to find some mutations there that would allow us to infer the subclonal hierarchy within the sample we're measuring, and then assign cells to their genetic subpopulations. And then we have both transcriptional subpopulations and genetic subpopulations. This actually technically turns out to be very difficult because, as you might imagine, coverage is extremely uneven, sparse, noisy. There's also this huge effect of monoallelic detection or monoallelic expression in some of the cells. So I actually use graphical models, but I'll skip that. I mean, the end point is that, from what we've seen, and again, we've only done this on a couple of tumor types, that every clone, actually genetic subclone, does have its own transcriptional signature that can be distinguished. But if you just look overall unbiased at transcriptional differences, the most striking differences do not appear to be genetic in nature. So they cut across subpopulation structure, that is, genetic subpopulation structure. And so, whether what relevance they have to the actual progression of the disease or response to treatment, that remains to be seen. But computationally, it's useful to have these types of methods that allow you to, group the cells within these tumors according to both transcriptional state, genetic state, and hopefully epigenetic state as well. All right, I'll just. You can close your eyes for a second. All right, just want to get to this slide. I want to thank my collaborators, so Joel Chun and Kuhn, David Skadden on the cancer projects, and also Peter Park and Woo-Young Park on this genetic transcription heterogeneity in cancers that I didn't go through in detail, as well as people in my lab, including Jean Fan, who's worked on this extensively. All right, thank you.